Our goal in this lab is to learn some techniques to analyze brain voltage data, which we can imagine were recorded in the scalp EEG. We will focus specifically on characterizing these brain rhythms using both visualization and other tools in MATLAB. There is much more to say about characterizing rhythmic brain activity, and what we'll do here is an extremely brief introduction to some basic techniques. In class, we discussed the idea of sine and cosine functions, which you've probably seen in other courses. To start, let's plot one of these functions and see how it behaves. So to start, let's define a time axis for our cosine function. We'll do this here in line 21. We'll define a variable t. It'll range from 0 to 1 in steps of 0.001. So what we're assuming here is that we're creating a time axis that starts at time 0, ends at 1 second, and takes a particular step size, in this case of 0.001. So let's define that here. We have this question, what is the interval between time points in this variable t? Well, we can see as we've defined t, it's 0.001, so we can think of this as 1 millisecond steps in our time axis. Let's now use this variable t to define a cosine function, and we'll do that in line 27. We'll define this new variable x. It'll equal cosine of some input. That input is 2 times pi times this t-axis. So let's define x here. So I do that. I can see in my workspace that I now have two variables, the time variable, which is 1 row by 1001 columns, and the x variable, which has the same dimensions, 1 row by 1001 columns. To visualize our cosine function, let's plot it. We do that in lines 31 through 33. So let's plot t and label our axes. We'll assume that the horizontal axis is time and the vertical axis is some signal. So I evaluate that and I get this plot where here we can see we have our signal, which ranges from 1 to -1, as we expect for a cosine or sine function, as a function of time, where here time ranges from time 0 all the way up to time 1. And if you look at this picture, can we decide through visual inspection alone what is the frequency of this sinusoid? Well, we can count the number of cycles that occur. If we start at a peak at time 0, we then move to a trough, and then back to a peak at time 1, so we complete 1 cycle in 1 second. That corresponds to a 1 Hertz oscillation. We can increase the frequency of this oscillation. Let's define a new variable f. We'll use f to stand for frequency. So I set f equals 4 here. And then let's redefine x. We'll define it again using the cosine function with a new input. The input is now 2 times pi times t. That's exactly what we had before, times this frequency f. So if we do this, so let's evaluate this new function x and plot it. And if I look at my figure, what do we have now? So if I look at this figure, what do we have? Well, here, if we look at the cosine function, it starts at 1 and goes to -1. So we have our signal on the vertical axis. We have time along the horizontal axis. Time still extends from 0 to 1 second. So here we're visualizing this x, and can we, through visual inspection, determine the frequency of this oscillation? Well, to do that, we can count the number of peaks that occur, the number of cycles that occur in this 1 second interval. If I start here at 1 and I go down to the trough and up to the peak, that's 1 cycle. I go down to the trough and up to the peak, that's a 2nd cycle. Down the trough and up to the peak, that's a 3rd cycle. Down the trough and up to the peak, that's a 4th cycle. So this oscillation completes 4 cycles in 1 second. That corresponds to a rhythm of 4 Hertz. Here we've only visualized a cosine function. We might also want to visualize a sine function. How can we do that? Well, let's define a new variable y, and let's set that equal to sine 2 times pi times t. So I've defined a new variable y. We're using our variable t, our time axis that we've already defined. I'll execute that here. And I see it has the same dimensions. This new variable y has the same dimensions as x and t. Let's freeze our graphics window and also plot this sine function. So here we'll plot t comma y, where now we're going to include this sine function on top of our plot. And what do we observe here? So we've added this new red curve. This is a sine function. How do I know? Well, I know something about sine functions. I can remember that they always have a value of 0, so the sine function has a value of 0 when the input is 0. Well, the cosine function has a value of 1 at an input of 0. So I can see this sine function here. How many cycles does it complete in this 1 second interval? Well, if we start here at 0, we then go up to a peak, down to a trough, and back to 0. So we complete 1 cycle in 1 second. That corresponds to 1 Hertz. You might wonder why I didn't stop here at time 0.5. So we start at 0, we go up to a peak and down to a trough. That's half a cycle. Right? This is only the upper part of the sine function. We also have to include the lower part to complete the full cycle. Now, in the second part of this lab, let's visualize some scalp EEG data. We introduced the idea of the scalp EEG in class. It's an electrode placed directly on the scalp surface of a human subject so that voltage activity is recorded as a function of time. Scalp EEG recordings are very common when doing maybe a clinical diagnosis of a patient with epilepsy or performing some experiment to understand how the brain voltage activity evolves during a particular task. So what we're going to do here is consider that we're working with a collaborator. This collaborator has provided us with a snippet of scalp EEG data recorded during an experiment and asked us to have a look at it and assess what rhythms are present. So let's do that. To download this data set, please go to Blackboard and download the file d1.mat. So I've done that. I've downloaded this file, d1.mat, and I'll load it into my workspace. So I load d1, and what I see is I have two variables that have been loaded into the workspace. This variable t and the variable v. And our collaborator tells us that the variable v corresponds to the voltage recorded at a single electrode, while the variable t corresponds to a time axis consistent with these data. Notice that I've cleared the workspace from part 1 of the lab, so I just have only these two variables, t and v, in my workspace. And as a good place to start, let's plot these data. So let's plot this voltage data provided by our collaborator. It's just this small snippet of data. So I've plotted it, and here it is. Here's the data that has been recorded from this human subject over some interval of time. There are some questions listed here in the lab, and maybe I would recommend that first you try to answer these questions on your own. Pause the video, look at these data, try to answer these questions, and then let's come back together and try to answer them in the video. So hopefully you've paused the video, you've thought about these questions, tried to answer them. Let's think about this first one in line 74. What is the total duration of the recording? It's hard to say here because I have not labeled my axes, so let's do that. Let's include this X label and Y label in lines 71 and 72, so I'll execute that. And now I've labeled my axes. What is the total duration of the recording? Well, I see that the recording starts at time 0, ends at time 8, so we have 8 seconds worth of data. Our collaborators provided us with these 8 seconds of scalp EEG data from a single electrode. Okay, so we know the total duration is 8 seconds. What is the time interval between points in the data? When I look at this data as is, right, it's hard to tell what the time interval is between points. Maybe I can zoom in and get a sense for the time interval between points. Right, so if I zoom in here on some interval of time between approximately 0.24 seconds and 0.4 seconds, I see the data is kind of jagged. Right? I see it jumps maybe from here to here, and then from here to here. So maybe if I zoom in even more, I can get a sense for what is the interval between samples. It's hard to tell because MATLAB is drawing a line in this case. So if instead I were to plot t comma v, and let's connect the points with a line and with asterisks. So each asterisk will indicate a single sample. Right, so here's the data again. I'll zoom in on some small interval of time. It doesn't matter which one. I've just chosen one at random here. And what we see is that we get a sample, looking at these data, we get a sample at 0.33 seconds, another sample at 0.34 seconds, another sample at 0.35 seconds, another sample at 0.36 seconds. So that suggests that the interval between these samples is 0.01 seconds. The time from 0.35 to 0.36 is 0.01 seconds. So we can see that by inspecting the data and plotting it in this particular way, where each sample is indicated by an asterisk and then MATLAB is drawing lines between these samples. So that's one way to deduce the time interval between points, is we can look at the data and try to inspect how far are these points separated along the horizontal axis. Another way we can do this is we can look at our time axis. So let's take t and let's plot indices from 1 to 10. And here they are. So these are the first 10 time indices in our time axis. And we see we start at time 0, we go to time 0.01 seconds, then 0.02 seconds, and 0.03 seconds. And we see that the interval between each one of these samples, again, is 0.01 seconds. So there's some piece of EEG equipment that our collaborator is using and is collecting the scalp EEG data every 0.01 seconds, or every 10 milliseconds. So that answers our second question. What is the time interval between points in the variable t? We looked at it graphically, and then we looked at this variable t by actually plotting the values, and we conclude 0.01 seconds. Okay, now maybe a more subjective question. This is line 78. What do you notice about these data? So what features do we notice about these data? Let's look at them again. So here's the data. I'll zoom out. I don't like this representation where we have all these little dots and asterisks, so I'm going to go back to the original plot where we only have a line. So let's plot t comma v. And here's the data plotted with only a line. This is a very subjective question. What do you notice about these data? Well, a couple of features that I notice are first these large amplitude variations. Right, there's some interval of time, maybe the first 2 seconds, we have these rapid fluctuations, maybe there's a slow trend in here, and then we have these faster oscillations occurring, and then suddenly we have these big deviations in the voltage. They look kind of sharp, right? These sharp downward deviations in the voltage. They last for some interval of time, from maybe 2.5 seconds to 7 seconds. We have these large deviations, and then the activity returns to these kind of fast fluctuations interspersed with a slower rhythm. Now on to the fourth question in line 80. What are the largest amplitude features in these data? Large amplitude, we're asking what are the rhythms that have the largest deviation in these data? And again, through visual inspection, it looks like whatever's happening from 2.5 to about 7 seconds, these sharp deviations in the data, whatever rhythm this is, appears to be the strongest. We have this sudden transition from these low amplitude fluctuations to suddenly this large rhythm appearing in these data. So I would say in this case, what are the largest amplitude features? Whatever's happening from 2.5 to 7 seconds. We might be able to approximate the frequency of this oscillation if I count the number of cycles that occur from 2.5 to 7 seconds. Again, very approximately. I count 1 cycle. I'm going to go from the min back to the min 2 cycles, the min back to the min 3 cycles, 4 cycles, 5 cycles, 6 cycles, 7 cycles. So approximately 7 cycles in approximately 5 seconds, from about 2 to 7. So we have 7 cycles in 5 seconds. That gives us a frequency near 1 hertz, maybe a little over 1 cycle per second. And that looks about right. If we look at the interval between these two troughs, or if we go from trough to trough, it's less than a second. I see it's between 3 and 4. So we have some rhythm happening here that's just over 1 hertz. And it's a somewhat strange rhythm where we have these sharp fluctuations, especially at the minima, and then these kind of smoother transitions at the maxima in this particular rhythm that we're observing. So we might go back to our experimental collaborator and ask, did something funny happen to this subject between about 2.5 and 7 seconds in this interval of data that we were given? Good, so we've thought about the largest amplitude features, these sharp, wiggly events that we see here from about 2.5 to 7 seconds. What are the dominant rhythms? Well, I think we've already answered that. We've looked at these large amplitude features. Those correspond to the most dominant rhythms in the data, the things that are easiest to see. And we characterize this rhythmic activity from about 2.5 to 7 seconds. We said there's about a little over 1 cycle per second. So some oscillation near 1 hertz, maybe a little over 1 hertz. There are other rhythms in the data that are not so dominant. We see these fast wiggles here, maybe in the first second. That's some other rhythm, but it's not the largest rhythm. It's not the one that stands out to the eye most obviously. So there are other rhythms occurring here, but the dominant one, I would argue in this case, are these large amplitude fluctuations that we observe from about 2.5 to 7 seconds. Now, in the third part of this lab, let's consider the power spectrum. In class, we discussed this technique. We introduced this idea of a quantitative method to assess rhythmic activity in EEG. There are many subtle issues to consider when you're applying power spectral analysis to real-world time series data. We won't cover those here, but if you do decide to dive into this area, be warned that there are lots of complexities to consider and also very fascinating problems. So to start, let's compute the power spectrum for the data D1 that we've loaded. To do that, please download this function powerspectrum.m from Blackboard, and we can look briefly at this function with particular focus on the 2 inputs and the 2 outputs that this function returns. So I've downloaded this function. I'll open it up here. Here is the power spectrum. I see that it takes 2 inputs. The first input, V, is the time series of interest, in our case, the voltage. and the second input, fs, is the sampling frequency. We'll have to figure out what that is, and we'll do that when we go back to the lab. So we need to define these two inputs, the signal, v, and the sampling frequency, fs. This function returns two outputs, p, the power, and f, the frequency. And to visualize the power spectrum, we'll plot p versus f, and we'll see that when we go back to the lab. Again, there are many different subtle issues to consider in computing the power spectrum. You might wonder, what are all these interesting functions like FFT? We're doing something here where we're computing 10 log 10, this decibel scaling. Why would we do that? Why is it useful? We then have to take half of the indices of the power spectrum. Why would we do that? And define this thing, the frequency axis, in a particular way. Again, there are many subtle issues and interesting details to consider here. If you're interested in it, please feel free to post to Piazza or email me, and I can send you a lot more information about what's happening here. But for now, let's just use this function, knowing that it takes two inputs, v, the signal of interest, and fs, the sampling frequency, and returns two outputs, the things we'd like to visualize, the power spectrum and a frequency axis. So let's go back to the lab and apply this power spectrum to our data of interest. So now I'm still in part 3 of the lab. I would like to compute the power spectrum and display it for our data. To do that, we know we need two inputs, v, the voltage, we have that signal already, and we need something called the sampling frequency. So how do we find the sampling frequency? We need the sampling frequency in order to display the power spectrum. How do we find it? Well, we can use our variable t in order to deduce the sampling frequency. So let's think about that. If we look at the first 10 indices of our variable t, I see that our time axis starts at time 0 and then goes to time 0.01, then to time 0.02, and then to time 0.03. So we see that the interval between samples is 10 milliseconds, or 0.01 seconds. To get the sampling frequency, we would like to determine how many samples occur in 1 second of observation. So again, the thing we would like to compute is the number of samples that we collect in 1 second of observation. Now, if we're getting a sample every 0.01 seconds, how many total samples would we get in 1 second? Well, if we take 1 second of observation and we divide it by the duration between samples, in this case 0.01, what we find in this case is we can fit 100 samples in 1 second of data. There are other ways you could determine the number of samples in 1 second of data. I've chosen one here. You might think of other ways of figuring out what is the sampling frequency for these data. The way I've done it here is the way I usually compute this quantity, and I think it's the most straightforward. You figure out the sampling interval, what's the time between samples, and ask how many of those intervals fit in 1 second of observation. Okay, now that we know fs, I'll define it here. So I know fs in this case is equal to 100. We're now ready to define the variable fs, our sampling frequency. To do so, let's first define the sampling interval, dt. That's the time between samples. Here I'll compute the time between samples as the time between the second sample and the first sample, so t2 minus t1. I've chosen two samples here, sample 1 and sample 2. You could try other samples as long as they're right next to each other, and you should get the same thing. So what do we find for dt? The time between samples in this case is 0.01 seconds, or 10 milliseconds. Exactly what we expect looking at our list of values above. Now with this, we're ready to compute the sampling frequency. We've said that the sampling frequency is the number of samples, dt, that we can fit in 1 second of data. So let's take 1 divided by dt, and what we find in this case is a sampling frequency of 100 hertz. So now that we know the sampling frequency, we're ready to compute the power spectrum. And let's do that. We'll copy line 118 from our lab. Let's compute P and F, the power and the frequency, by calling this function power spectrum, where we're going to input our data V and our sampling frequency Fs. So I'll compute that here. It doesn't crash. Good, I'm in the right directory. This function seems to work. And now let's visualize the results. Let's plot the power versus the frequency. So I'll do that here. Let's label the axes. So I'll put an X label in here for the horizontal axis. Let's make that frequency in hertz, and then in the horizontal, or on the vertical axis in the Y label, let's label that power in decibels. And now what's interesting about this power spectrum is we can inspect it and determine what rhythms dominate. Now what do we mean by that, finding the rhythms that dominate? Well, what we'd like to do is isolate the rhythms that have the most power in our signal. So we're looking for peaks in this power spectrum. What are the frequencies on the horizontal axis that have the highest power on the vertical axis? And I notice, maybe the first thing we notice is that this power spectrum is very spiky, right? There are lots of up and down sharp parts in here, but most of the power is concentrated at low frequencies, especially frequencies lower than 10 hertz. So let's focus on this region here, these frequencies less than 10 hertz. So I'll zoom in on that region. I'll try to isolate this interval between approximately 0 and 10 hertz. And what we notice is that there are a couple of interesting non-zero frequencies. So I won't focus on the zero frequency component. That corresponds to the mean. Not interesting for us. What is an oscillation with zero frequency? It's something that's not wiggling at all. And we're interested in the wiggles in the brain, in this case, the oscillations of non-zero frequency. And in this interval, from 0 to 10 hertz, the largest peak, the peak with the most power, occurs at a frequency between about 1 and 2 hertz. So it's this peak right here. So how do we interpret this peak? And is it consistent with the data that we initially observed? Well, remember, when we plotted the voltage versus the time, what struck us, and maybe the most dominant feature, were those large blasts of activity. And we found that the interval between those blasts was slightly less than a second. And if we had to approximate the frequency, we would have said those blasts occurred between about 1 and 2 hertz. And that's exactly what we're finding here, is a peak in the power spectrum between about 1 and 2 hertz, which corresponds to the largest amplitude activity we observe in this signal, those large amplitude blasts. So in this way, the power spectrum is characterizing the rhythmic activity in the signal. It's telling us the signal has a dominant rhythm, and it has a frequency between about 1 and 2 hertz. And again, this is consistent with our visual inspection of the data, where we notice these large blasts of activity with a frequency that we counted by hand of approximately 1 to 2 hertz. Now, in the last part of this lab, part 4, let's analyze another EEG dataset. To do so, please go to Blackboard and download the file d2.mat. So I've already done that. This is another snippet of data that our collaborator has provided us. So I'll load d2.mat into my workspace, and I see that 2 variables are provided. Just as before, we have the variable v for voltage and t for time. And we'd like to analyze these data and try to make sense of any rhythmic activity that occurs. As a good place to start, let's get our bearings by computing 2 quantities. What is the duration of the recording, and what is the sampling frequency? To determine those 2 quantities, let's look at the variable t. So if we look at the variable t, at what time does it start? So recording starts at time 0, and our collaborator tells us this is time 0 in seconds. When does it end? So time goes from time 0 all the way up to time near 4 seconds, in this case 3.99 seconds. So we have approximately 4 seconds worth of data, this snippet of data that our collaborator has provided us. To determine the sampling frequency, we need to first determine the time between samples. So we might do something like look at the first 10 samples. We see the first sample occurs at time 0, the next sample at time 0.01, the next sample at time 0.02, and so on. So here we see that the time between samples is 0.01 seconds. We can define a variable dt. Let's set that equal to the time between samples. We might compute the time between the second and first sample, and I get a value in this case of 0.01, or we could have set dt is equal to t of 10 minus t of 9. So again, this is the time between 2 samples, where I've chosen the 10th and the 9th sample, and I get the same thing. The sampling interval is regular. It doesn't matter which 2 samples we choose as long as they're next to each other. So we find that the sampling interval, 0.01 seconds. To compute the sampling frequency, we take 1 over the sampling interval. And that, in this case, it's 100 hertz. So we have 100 samples per second, just like the first data set, D1, that we analyzed. So now that we have these first 2 interesting quantities, let's visualize the data. Always a good place to start, so let's plot the data versus time. And so here's a plot of the data. Let me label the axes. So we'll label the x-axis as time in seconds, and the y-axis, let's label that as voltage. And so here we have our figure with the axes labeled. We have 4 seconds of data, and here we're plotting the voltage, in this case in volts, on the vertical axis. And what we'd like to do is to start, just look at these data and determine, are there any rhythms that stand out? This is a very complicated data set. And the reason I'm showing it to you is this is what real human data, this is what brain data looks like. It's complicated, and typically rhythms don't just jump out at us. What we can do is maybe we'll zoom in a bit. So I'll try to zoom in on these data, and let's see if we can find anything that looks like rhythmic activity and just get a sense for the type of rhythms we observe here. So I'm going to zoom in between about 1.5 and 2 seconds. Do we see rhythmic structure here? Well, maybe we do. We see something that goes from a peak to a trough, back to a peak. So there's one cycle, a peak to a trough, back to a peak, another cycle, peak to a trough, back to a peak, another cycle, peak to a trough to a peak. There's another cycle. So maybe we're seeing something rhythmic in here. Let me zoom in again. Now here I'll zoom in between about 1.8 and 2 seconds. So I'm zooming in between about 1.8 and 2 seconds, and we could try to approximate the frequency of this rhythmic activity. So what do we have? So we're starting with our peak in our rhythm at about 1.82 seconds. We go down one step to 1.83 seconds, 1.84 seconds, and we go back up 1.85 seconds. So the time from peak to peak is from 1.82 seconds to 1.85 seconds. So that's 30 milliseconds, or 0.03 seconds from peak to peak. What's the frequency? Well, if we take 1 divided by 0.03, we get about 33 hertz. Let's compute another cycle in these data. If we start at the next peak, 1.85 seconds, we move forward 1 sample, 2 samples, 3 samples, 4 samples, we get to the next peak. So here we've gone from peak to trough to peak. The first peak was at 1.85 seconds, and the next peak was at 1.89 seconds. So we can compute the difference, 1.89 seconds minus 1.85 seconds, and we find that the interval between these two peaks, in this case, 0.04 seconds, and that corresponds to 25 hertz for this cycle. We can move on to the next peak. Let's try one more. We go from peak here at near 1.89 seconds all the way to the next peak at 1.93 seconds. So again, we go peak to trough to peak. There's one cycle. If we take the difference, 1.93 minus 1.89, we again get 0.04 seconds. So we find another 25 hertz cycle. And we can repeat this procedure. We can keep computing the time from peak to peak, counting up the interval, the duration between these two peaks, and then computing 1 over that to figure out the frequency of that single cycle. And if you do that over this interval, you'll find you get frequencies between about 20 hertz, maybe all the way up to 40 hertz. So it seems like whatever rhythmic activity is happening here, it's occurring over a broad range of frequencies. Let's now compare this observation to the power spectrum. So we can compute the power spectrum for the data. Let's do this. We'd like to compute the power and frequency. We'll call this function power spectrum, and we'll send in our data, which is V, and we have the sampling frequency fs, in this case 100 hertz. So I'll call that function and let's visualize it. Let's plot f comma p. So we'll plot the power spectrum versus frequency, and there it is. And let me label the axes. So the horizontal axis will label frequency in hertz. And then the vertical axis, the Y label, will label that power in decibels, a particular scale for examining the power spectrum. And here we have the power spectrum for these data. What do we notice about it? Well, the first thing we might notice is that, again, it's very jagged. This looks like real human power spectra, where you have lots of different peaks and troughs. It's kind of hard to see exactly what's going on. If we try to isolate peaks in these data, maybe we would isolate a peak here at very low frequencies. So maybe we should go back and see if there's something happening between 0 and 5 hertz in this case. What's happening at this peak? But the thing I'd like to draw your attention to is this rather broad peak. So if you kind of squint your eyes and look at this, you might notice this broad elevation. All right, so the power spectrum starts high, then goes down, and then we have this broad elevation in power between about 20 hertz and 40 hertz. Maybe that peak does not stand out. You might imagine smoothing this curve, get rid of all these little jagged wiggles, and just smooth this curve. And perhaps if you can imagine fitting a smooth curve through here, you can see this slight elevation in the power between about 20 and 40 hertz. And perhaps the most important thing to notice here is that this broad peak is consistent with our visual inspection of the data. We looked at 3 cycles and we found frequencies of 20 hertz and 33 hertz, and those lie within this broad extension in power. So at least our initial visual inspection of voltage versus time deduced the same rhythms that we're seeing here in our power spectrum. They're high frequency between about 20 and 40 hertz, and they occur over a broad range of frequencies. The reason I like this example is that it's consistent with the types of power spectra you see in actual neural systems. It's very unlikely, or it's very uncommon, to see isolated peaks at very narrow frequency intervals in the power spectrum. Instead, when you analyze brain activity, you often see these peaks that have broad extensions over frequency. And that's exactly what we see here. We had this broad peak from about 20 to 40 hertz. Remember, when we were talking about frequency bands, we talked about an alpha band, 8 to 12 hertz, or a theta band, 4 to 8 hertz. These are bands of frequency, not individual frequencies. So when we talk about the alpha band, we don't talk about a fixed 10 hertz oscillation. Instead, we talk about a range of oscillations between about 8 to 12 hertz. Again, that's very common in neural systems, and that's exactly what we're seeing here. So although this is a complicated example, again, I think it's representative of the types of data you see when you try to analyze real neural systems. In the assignment this week, you'll be asked to analyze additional data sets, see if you can extract the interesting rhythms that occur, and you'll also be asked to look at some power spectra extracted from papers and see if you could say anything about the rhythms that occur based on the power spectra that you see.